organization of cytoskeleton in appropriate manner is very important for cellular functions. Actin filaments can be organized in two different ways. Either actin filaments can be brought together in the form of bundles, and there are two types of bundles. I'll tell you about them. Or actin filaments can be linked in such a way that they form these cross mesh-like structure two-dimensionally, or in three dimensions. If they're linked in three-dimensionally, they can form these gels, and they give cells different properties. So let's talk about proteins that organize actin filaments into these different orientations. Actin filament cross-linking proteins are divided into two classes: bundling proteins and gel-forming proteins. Bundling proteins cross-link actin filaments into parallel arrays, while gel-forming proteins hold two actin filaments at a larger angle to each other, creating a loose meshwork. Both types of cross-linking proteins generally have two similar actin filament binding sites, which can either be part of a single polypeptide chain, or it can be contributed by two polypeptide chains held together in a form of a dimer. I'll talk about both these categories. I'll give you examples of them, and I'll also show you these two different categories of proteins that can link actin filaments. Spacing and arrangement of these two filament binding domains determines the type of actin structure that a given cross-linking protein will form. Here, let me show you this diagram. In this diagram, you will see that we have different organizations of actin filaments. In stress fibers, for example, you have these actin filaments arranged at a specific distance, and this distance, if you would notice, is greater as compared to the distance between actin filaments in filipodium. Additionally, if you would notice that polarity of these actin filaments is mixed. The arrows basically point towards the plus end. Certain actin filaments have their plus ends towards the cell periphery. Others have their plus ends towards the cell center. The tight bundles found in Filipodia are all parallel, and they have their plus ends pointing in the same direction. The tight bundling of actin filaments give Filipodia a greater structural support, whereas the contractile bundles, the relatively sparse spacing of actin filaments allows motor proteins, we'll talk about motor proteins, for example, myosins, to insert themselves between the actin filaments. And once these motor proteins are activated, they result in overall contraction of these bundles of actin, resulting in change of cellular shape, or they can also be used to, for example, stretch adhesion molecules that we talked about when we were talking about adhesion, cell adhesion. This is a crawling cell. This cell can move. That's why it has these Filipodia. Filipodia basically also serve a function of little fingers. They can sense the environment and tell which direction cell should move. Also, in the cortical actin, which is found near the membrane, this area, the actin filaments in this area, they form this gel-like network because they are not oriented parallel or anti-parallel to each other like the other two categories we looked at. And this makes this region a little bit more viscous, and also this type of actin arrangement is generally found in lamellopodia, which allows cells to stay stuck at a particular location and also keep the cell shape. Now let me show you some of the proteins that help actin organize themselves in a specific manner. Fibrin is a small cross-linking protein, and this protein has two actin binding domains close together in a single polypeptide chain. I would like to point out that fibrin and alpha-actinin, I'll talk about that in a minute, are widely distributed actin bundling proteins which can align actin filaments. Fibrin is enriched in parallel actin filaments in filopodia that we saw in the diagram on the previous slide at the leading edge of the cell and is responsible for tight association of these actin filaments. I'll talk about alpha-actinin and then I'll show you that image again, that diagram again, so you can appreciate what these proteins are doing. Alpha-actinin contains two actin binding domains that are further apart, and it is concentrated in stress fibers, where it is responsible for relative loose cross-linking of actin filaments, and also it helps, alpha-actinin also helps form structures that form the stress fibers in the focal context at the plasma membrane. So keep this in mind. This is here we have alpha-actinin. It's a dimer. The actin binding domains of this dimer are relatively further apart as compared to the fibrin. These red squares are the actin binding domains. Also notice that since the alpha-actin dimer is antiparallel, head of one alpha-actinin protein is adjacent to the tail, if you will, of the other alpha-actinin protein, and together they're forming this dimer. So consequently, since they are themselves in anti-parallel organization, configuration, when the actin filaments bind, they will bind them in such a way that they are also themselves anti-parallel. Now let me take you back to the diagram we looked at in the previous slide. So here in stress fiber, you will need a protein which can hold actin filaments at a relatively greater distance, which is as we have seen is a job done by alpha-actinin. Whereas tight bundles, we would need a linker protein or a protein that can grab actin filaments closer together, and that we said was a function of fibrin. Each type of bundling protein determines which other molecule can interact with actin filament. Myosin 2 is the protein in stress fibers and other contractile arrays, and that is responsible for their ability to contract. The very close packing of actin filaments caused by fibrin excludes myosin, therefore filopodia are not contractile. The looser packing caused by alpha-actinin allows myosin molecules to enter and consequently making these stress fibers contractile. Different spacing between actin filaments, for example, bundling by fibrin automatically discourages bundling by alpha-actinin and vice versa. So the two types of bundling proteins are by themselves mutually exclusive. If you have fibrin, the alpha-actinin won't be able to bind those actin filaments and vice versa. Here, this diagram shows you relatively how these two actin bundling proteins are holding actin filaments. So again, the orientation of actin filaments bundled by alpha-actinin are antiparallel and they're further spaced apart compared to the protein fibrin, which also bundles actin filaments. In this case, as you can well imagine, you would not be able to place myosin motor protein in between these filaments, whereas in this case, you have enough room to have myosin between the actin filaments. So in the next module, we'll talk about other proteins, actin-associated proteins that can hold actins, form bundles, and also we'll talk about proteins that can form the gel-like meshwork we talked about.